Okay, so let's go. The last talk of this section is my long-term friend, Dr. Liu Shih-Jen. Dr. Liu is the PhD in the MIT. Okay, graduate, got his bachelor degree in the Department of Pharmacy in Kaohsiung Medical College, and he got his PhD in National Defense Medical Center. After the 2 years the military service, he did join the biotech company for 5 years. Then he became the assistant investigator vaccine R&D center at the NHI in 2006, and then promoted to the full investigator in 2015. And then he spent 5 years to become the CEO of bioproduction plant called the CGMP facility at the NHI. So currently Dr. Liu is the associate director of his institute. I know that Dr. Liu for many times, and his research is more applied research, so that's why he won several times the National Innovation Award and the Future Science Technology Breakthrough Award. So let's welcome Dr. Liu.
Thank you very much for the Jingbang kind introduction. It's my honor to present our data here and I share our research. So today I would like to talk about the preclinical development of the clinical of the DNA vaccine. So the DNA vaccine, I summarize the past clinical trial in the clinical trial government website, and you can see the DNA vaccine clinical trial around hundreds, many hundreds, and most of the clinical trial in the phase 1 and phase 2, and some of the move to the phase 3. And the DNA vaccine almost more focused on the HIV and the cancer, except the COVID-19. So why using the DNA vaccine to apply in the HIV therapy and the cancer therapy? Because the DNA vaccine can induce both the CD4 T cell and the CD8 T cell immune response. So DNA vaccination, so the DNA can produce the protein in the cytosol and go through the proteasome degradation and move to ER. And go through the MHC presentation pathway and activate the CD8 T cell. And also they can secrete the protein, and the protein uptake by the antigen presenting cell and go through the MHC class 2 presentation pathway and activate the CD4 T cell. So that's why DNA vaccine were used in the cancer therapy and the HIV therapy. So the DNA vaccine, based on this the delivery method, we can divide it two categories. One is live vector delivery. The other one is chemical and physical delivery. The live vector delivery, you can deliver the DNA using the bacteria or using the live virus. And most of people have been vaccinated using the AZ chimpanzee adenovirus vaccine for COVID-19, and also the Johnson & Johnson using adenovirus 26. Okay, and today I would like more focus on the chemical and the physical delivery, especially using the electroporation. And this one is Zycov is produced in the Indian and got the EUA in Indian. So the design DNA vaccine, the first step is identify the antigen, and then you have to optimize your plasmid. You may gene codon optimization, codon sequence design, or leader sequence, and so on. And you also need to design using your plasmid formulation with the liposome, microparticle, or nanoparticle, or aluminum. Or you can deliver it using different methods, like a gene gun, pressure injection, electroporation, microneedle, and also the pharmajet, topis, and so on. So that can induce the immune response in human. So I just talk a bit of our COVID-19 DNA vaccine development. So we using the DNA vaccination with the electroporation. We compare the in IM and the electroporation and without the electroporation. So you can see here after electroporation, the antigen expression is very high than the non-electroporation. That means the electroporation can efficiently induce the protein expression. That can also induce the beta immune response. So we, in the COVID-19 outbreak, so we start to design different truncation of the COVID spike protein using different design, maybe the full length or RBD only, or RBD and the transmembrane domain, and the transmembrane deletion. And here is the protein expression to confirm the contrast. After that, we start to immunize in mice and evaluate the antibody titer. The first one is the anti-spike antibody titer. So all of the construction can induce the good antibody titer against the spike protein. And we also test the anti-RBD region. So here, only the RBD can induce the beta RBD antibody response. The most important is how about the neutralizing antibody titer. So we compare the different contra and compare the neutralizing antibody titer. And we saw the RBD alone and the full length both induce the beta neutralizing antibody titer. So we started using these two contra to immunize the hamster and to see the protector effect. So we immunize the RBD. The blue one is the RBD, and the red one is full length. So you can see the full length and the RBD plasmid immunization can induce the antibody production, but only the full length can induce the beta antibody immune response. And also in the neutralizing titer, also only the full length induce the beta neutralizing antibody, but not the RBD alone. So that's a very interesting result. So we also test the challenge with the virus, and we monitor the body weight. So the full length plasmid can protect the animal from the body weight loss. And also we analyze the viral load in lung, and the full length plasmid immunization can reduce the viral load in lung. Okay. So here it says, have a result showing the DNA vaccination with the electroporation can induce protected immunity in animal. And we also use a similar approach to again to do the Zika virus challenge. And we designed the pre-M and the E, the antigen. We select this antigen and use a different dose to see the neutralization titer. And you can see the 40 microgram can induce very good neutralization titer. And also after challenging the 40 microgram and the 20 microgram can induce the survival rate around 60 to 70. And here just showing the viral load after virus challenge. So higher dose of the DNA vaccination can protect the animal from the virus challenge. And we also compare this method to the mRNA and LNP in the H7N9 influenza virus. We're using 2 micrograms of the HA DNA or mRNA of the H7N9. And after 2 vaccination, we challenge you with the virus, and then you can see the DNA and the mRNA and LNP both can produce 100% survival. That means the DNA electroporation is comparable effect to the mRNA and LNP. So we think that's very important for the DNA vaccination. But all this data is using in the animal. So how can we apply our result in the human? That's the challenge, because using the electroporation in animal is simple. However, how to apply this in the human? So that is our major challenge. Okay, so in the COVID-19, actually, we started DNA vaccination. We also contact the electroporation machine in the US to find a possible collaboration, but none of the company have response to us. So we think maybe we have to use our technology using the electro-acupuncture machine. And we design a syringe holder with disposable needle set. So this needle set is disposable, one by one you can use, okay? But the here design maybe is simple, but how can this design can induce protective immunity in human, in animal? So we start to design this machine and using to immunize. So because the machine limitation is below the 40 voltage, so we from the 5 to 40, and all this voltage can induce very good antibody titer, and also induce very good neutralizing antibody titer. And we also detect the injection site to see any safety issue or any concern. We can see the injection site is all of the recovery, minor damage. And we also compare the electro-acupuncture machine induce the T-cell immune response. We compare the plasmid with the electroporation or without the electroporation, and also compare the recurrent protein formulated with alum. And this formulation is for the Th2 immune response. So you can see the plasmid using the electroporation can induce a very high level of interferon gamma, and the recombinant alum, recurrent protein and alum formulation induce higher level of the IL-5 and the IL-13. So the level of the interferon gamma and the IL-5 ratio or interferon gamma IL-13 ratio all demonstrate the DNA vaccination with the electroporation induced Th1 immune response. And also similar data can using the interferon gamma spot have the similar result. So we also using this approach apply into the hamster challenge model, and also we compare the non-electroporation, and you can see here anti-spike protein antibody titer 5 voltage or 40 voltage have the similar level of the antibody titer, neutralization titer, and after challenge they can reduce the viral load in lung, and also their body weight loss is not observed. But if you don't have the electroporation, the body weight is loose. And we also have the histologic, analyze the tissue of the lung and see the inflammation, and the 5 and the 40 voltage electroporation have protective effect. So that means this using the electropuncture machine deliver the DNA vaccine can induce the protective immunity in animal. So after that, we start to GMP production of our plasmid and to send out the lung to do the non-clinical toxicology study. And we using the SD rate and IM injection, follow the electroporation once every 2 weeks, and we checked all the parameters that the regulatory acts we have provided. And we also do the biodistribution and the persistence of the DNA plasmid following 2-dose vaccination in rat. And because DNA vaccine have been developed for many years, so the regulation is very clear. The injection, after injection at the 6th day, you cannot detect the DNA copy number more than 30,000 per microgram. So after immunization, we check all the tissue and this organ, and we found none of the tissue or organ have the DNA copy number more than 30,000 copy per microgram. So here is an injection site. Most of the DNA plasmid is in the injection site and the giant lymph node, not other organ. So this data are submitted to CDE. So in summary, our GLP preclinical toxicology is performed by the local CRO company. There are no significant toxicity was observed in this study. And the result have been reviewed with the CDE, and no further concern or safety concern were raised. And the DNA vaccination with electroporation machine is feasible approach for emerging infectious disease outbreak. And the GLP preclinical toxicity and the GMP production of the DNA is finished. The application for the IND has been approved by the TFDA this year. So how about it? Because now COVID-19 is not a major threat in our society. So how about to apply this DNA-based therapy in cancer? That's the next stage. In our previous data, we designed a DNA vaccine have N-terminal, have a ER targeting sequence we call the E319K. So using this ER targeting sequence and have antigen. In that time, we using the HPV 18, E6, E7. After DNA vaccination, we can induce a CTL response. And this is immunized with the HLA A11 transgenic mice and induced, and both we identified two peptide is the HLA A11 CTL epitope that can induce CTL response. That means using this design can induce the CTL immune response. So we start to think about how to apply this in the tumor-associated antigen. So we using the tumor-associated antigen at 6 and design the different construct. And one of the construct is using the ER targeting sequence linked with the tumor-associated antigen at 6. And you can see this is a colocalization of the antigen and in the ER. So here is the colocalization, and here is the data, statistical data. So TAL6 is really located in the ER, most of the expression protein. So after that, we also evaluate the CTL immune response. So here you can see the leadership ER targeting sequence L6 DNA vaccination induce higher level of the CTL immune response compared to the non-ER targeting sequence. And also the antigen can induce the specific interferon gamma secretion. And the A2-5 is one CTL epitope we identify. Okay, so you can see here, induces a very high titer, high level of interferon gamma. And also we test the antigen-specific CD8 T cell using the tetramer technology, and also can detect the antigen-specific CD8 T cell is higher in the ER targeting sequence TL6. Because TL6 is a surface protein, so we also measure the antibody-dependent cellular cell toxicity, and both the P-L6, both L6 construct can induce the ADC effect. That means this design can induce both the CTL and the ADCC. And this model not only can induce CTL, they also can inhibit the tumor growth in EL4 lymphoma and also B16 melanoma in these two tumor models. So here is very clear. If you did not have the immunization with the L6 plasmid, they have a lot of melanoma in lung, and here is after we treat it with the L6 and the EK-L6. So how about the DNA vaccination in neoantigen-based cancer therapy? This may be the future application in this technology. So we find a paper published in 2020. They have using the CT26 and using the neoantigen from the CT26, and after vaccination, and also combined with the anti-CTLA-4. But in the plasmid without the combination, they don't have any tumor suppressor effect. However, if the neoantigen plasmid combined with the anti-CTLA-4, they can induce a very good tumor suppressor effect. That means this DNA vaccination approach combined with the IO, maybe the very good approach to the cancer therapy. And we also using this sequence and combine our E390K technology, and we treat the CT26 tumor at the day 3 start the treatment. So we also can find they can inhibit the tumor growth. But that means the E390K leader sequence can enhance the CTLA response and also inhibit the tumor growth. Okay, so I will summarize here is the DNA vaccination is strong protective effect in the SARS-CoV-2, and the DNA-based cancer vaccine can be used for in combination with the future with the immune checkpoint inhibitor. And the DNA vaccination with electroacupuncture machine can be used in human to investigate therapeutic efficacy of the cancer vaccine. So I will stop here, and I'm happy to take any question from you. Thank you very much.
Please. Your electropunctuation device that has been done before, but not quite successful because the yield is not high enough. So could you elaborate more? Are you going to how you try to conquer this issue?
Okay, actually, using the acupuncture machine, not any acupuncture machine could be used in this DNA vaccination. Actually, we compare the wave and the frequency and compare the different things. So that's we selected one from the different acupuncture. They have different brand of the acupuncture machine, but we select what we want because we're using the electroporation in the animal. And we using the similar web to do the experiment. So I think that's the difference. Yeah.
Very nice talk. Thank you. Just thinking ahead, when you imagine talking to regulators like Taiwan FDA, other FDA, EMA, I wonder if they were going to think about this as a combination product, device plus drug. Have you had any thoughts about that? Because sometimes that's a bit challenging.
Yeah. Actually, the combination, they need to evaluate your product and the machine together. So you can see the FDA not only evaluate our DNA vaccine quality, they also evaluate the machine quality and also need to do a lot of the machine to demonstrate that the machine is safe in animal. So I did not talk many because that's a lot of work. I think you know that. That's a lot of work to let the FDA allow you to do the phase 1 clinical trial for the IND. Yeah.
I can tell you're very brave to go down the combination pathway. Congratulations.
Thank you.
A very quick question. I have lots of questions, but just one simple question.
We can
so
talk.
you use E6, E7, for example.
Yeah.
So E6, E7 are oncogenic proteins.
Yeah.
Did you use a part of the protein? Or
Full length. E6
length?
full length and the E7.
Is there any risk to induce the cancer if you use an entire E6 or E7?
Because we using a mutation one. Because we mutate the oncogenic side.
I don't understand well, but I think it's risky. And for the last one, you combined with the anti-CTLA-4 antibody, not anti-PD-L1 or PD-1. So is there any reason you use a combination with the anti-CTLA?
That paper is part of other group, not ourselves.
Okay.
Right? Because we just start this project. So our result is the last one. We only the neoantigen fuse ER targeting sequence.
So
that's our unique.
How many neoantigens?
20.
20,
Yes.
just for the class 1, not
Class O and the class 2, yes.
class 2 as well.
Yes,
Okay.
well, yes.
So we have more time. We still have the 3 minutes. So is there any question? No? Okay, so let's thank you again for Dr. Liu.
Thank you very much.